Hello everyone. Welcome to course Recombinant DNA Technology. Today we are going to talk about Introduction to Recombinant DNA Technology. First, we have the concept of gene recombination. Okay. So the thing is the recombinant DNA technology which defines the gene recombination. Okay. And if you see the definition, which is transfer of genetic information from one organism to the another organism through manipulation of genetic material to get desired goal. And present day RDNA technology has its roots in the experiments performed by Boyer and Cohen. Basic principles of RDNA technology. So here we have generation of DNA fragments and selection of desired piece of DNA. And then you have insertion of selected DNA into cloning vector to create rDNA or chimeric DNA. And we have introduction of the recombinant vectors into the host cell. And also we have multiplications and selection of clones containing the recombinant molecules. And finally, we have expression of genes to the produce to desired product. For example, so this is the diagrammatic structure which tells us how the restrictions and ligations are made and then recombinant molecule, how it is produced. So this is the DNA 1 and this is the DNA 2. So this DNA from one sources, this DNA from the another sources. So this both DNA molecule has been restricted by using ECO1 enzyme, and once you restricted and you'll be getting sticky in nature of DNA molecule. So this DNA, both DNA molecule will be ligated through DNA ligase enzymes. Once it is ligated, then you will have the recombinant DNA molecule or chimeric DNA. So this is the perfect picture which will tell us the production of recombinant DNA using human and bacterial plasmids. So as we know, many were affected with the diabetics disease, that is the development of diabetics in later stage of their age. And once this insulin gene, which is functional, is restored or normalized, in that case, the continuous production of insulin will be occurring. Once it is occurring, and then you will start to control over the glucose molecules. Okay, so here we have the insulin gene, which is isolated from the human chromosomes. This insulin gene has been restricted to get only the insulin gene, not any other genes. And then, so we have the plasmid DNA molecule. So this plasmid DNA, bacterial plasmid DNA molecule also restricted with the restriction enzymes, and both are ligated. Once this ligated steps is over, so here we have the ligated steps, and then we will be transforming into the host cells. So this host cells will try to express this recombinant bacterial plasmid and will started to produce the insulin continuously. And finally, we can able to get the insulin protein through this bacterial cells. The first thing, the restriction endonucleases. So this is the enzymes which can easily manipulate the DNA, and also these enzymes which is obtained by from the bacterial enzymes, bacterial cells, and these enzymes are the part of bacterial enzymes. This can specifically cut or split the DNA at specific sites. And so this particular restriction endonuclease enzymes were first discovered in E. coli and restricting the replication of bacteriophages by cutting its viral DNA. So this mechanism will enable the bacterial cells to prevent the bacteriophages or the viral particle to replicate it again and again and destroying the E. coli cells, that is the bacterial cells. And through this, this equally DNA also be protected from the cleavage of its DNA by using methyl group. So this particular virus particle will be actively going to bind to the E. coli DNA, and this E. coli DNA, once it has the methyl group in its DNA, then it can able to easily prevent the digestion or restrictions. And so these enzymes also restrict the viral replications, are known as the restriction enzymes or restriction endonuclease. So before going into the other parts, like we can um, think about other restriction enzymes. So there are two restriction enzymes. The first one is the restriction endonuclease, another one is the restriction exonuclease. And this restriction endonuclease category will be preferred frequently in molecular biology experiments because it has significance in middle cleaving of middle DNA. If it is anything we are going to specify it, so that part alone will be cleaved. And another important thing, recognition sequence. So this recognition sequence, this site where the DNA is cut by restriction endonuclease enzymes. So this recognition sequence will be read by the restriction enzymes in DNA molecule, and it can specifically make the recognition and the splits or the cleavage. The ranges would be 4 to 8 nucleotides. And cleavage pattern, this is the another important thing. So cleavage pattern, so this generally these restriction enzymes can able to make sticky end or blended nature of a cleavage. So this sticky end nature of cleavage, which is very useful for RDNA experiments, and many are preferring to take up only the sticky nature of RDNA molecule rather than the blended cleaved DNA molecules. And if someone is considered the blend-in, and still they need to go for other adapters or linkers, other molecule support to refine their structure more precise and to have more affinity towards the plasmid. Okay, so in that case, the linkers and adapters are very important if you consider the blend-in nature of cleavage DNA molecule. So here we have restriction enzymes types. For example, here this type 1, type 2, type 3, and also we have type 4. And this type 1 will have the digestions and also which do not generate specific fragments. That is more important. So we were talking about the recognition sites or recognition sequence. So in that, if the template DNA is there, in that case, if this type 1 restriction endonuclease enzymes, which is going to identify a particular sequence and make a cleavage at some other sites. So it can't recognize at particular sites, and also it won't make the same recognition split up in the same recognized site. And this particular type 1 restriction endonuclease enzymes, which needs magnesium ions and ATP S adenosyl methionine for activating their enzymes. And also it can able to track the DNA for variable distance before breaking. That is the away from the specific recognized sequence. And type 2. So this is the very important restriction endonuclease enzymes, so which makes the recognition and also make cleavage at particular site. So it won't generate the cleavage away from the recognition site, so which will precisely identify the particular sequence and also make the restriction on same site. Okay, that is very important for molecular biology or DNA technology experiments. And so this type 2 restriction endonuclease enzymes, which makes the specific sequence with the help of magnesium 2 ions, and also which doesn't need ATP or S adenosyl methionine for activating their enzymes, and also it doesn't need, that is the ATP and So as usual, we have the adenosyl methionine. So this is doesn't need here. So only one um, part, that is the subproduct which is needed for activating the type 2 enzyme, that is the magnesium uh, 2 plus ions. And type 3 we have, so this particular enzymes which needs ATP as adenosyl, but which doesn't needs the magnesium ions. And this particular type 3 restriction endonuclease enzymes which makes the break away from the recognition sequence, which would be around 25 base pairs away. So these are the examples. So if you see the example here, so we have the list of microorganisms here. So this list of microorganisms which can able to produce this sort of enzymes, that is the restriction endonuclease or exonuclease enzymes. And this is the restriction sites. So this particular sites can be easily identified through this restriction enzymes and make a cleavage. And here you can see the first one, BAM-H1, which will make the sticky end. The second one, BAL, which will make the blend end. And third one, ECOR1, which will make the sticky end. Likewise, the enzymes will vary in their pattern and also which can have different size of nucleotide restriction. And we have the symptomatic diagrammatic representation for ACOR1 restriction enzymes. So this is the, if you consider, so this is the first ACOR1 template DNA. Okay, so for example, this could be the plasmid DNA. So both DNA molecules, the source can be restricted by using ECOR1. Once it is restricted, and which will have the sticky in nature. And here you can able to give the DNA ligase enzymes. Once it will try to heal both strands, that is the both source of DNA sticky and molecule can be ligated. Once if you ligated, and which will give you the recombinant DNA molecule through ECOR1, that is the ECOR1 which has the significant role here, and the DNA ligase enzymes which also has an important role here. So this is the another diagram, so which explains the blend in nature and sticky in nature of restriction endonuclease action. So this is the ECO1 again. This is the template stand. Here we have the template stand. So this template stand, which can be again um, can be restricted by using ECO1. Once this ECO1 is identifying the specific sites, and it will make the cleavage, and which will give you the sticky in nature. Whereas the another enzymes called SMA, that is the SMA1, so which will give you the blended nature of products. So here we have same example for blendings. So here you can able to see that, but it doesn't have any nucleotides codes here. So that's why you have as general diagrammatic representation here. And this is the another important thing, DNA ligase enzymes. So this ligase enzymes were originally isolated from the viruses, also occurs in E. coli and also in few eukaryotic cells. These DNA ligase enzymes, which are covalently joined together by DNA ligase enzymes. Okay, so for example, if the DNA fragments be restricted, if we get the DNA fragments through the restriction enzymes, and those restricted fragments can be easily ligated through the covalent bonding nature by this DNA ligase enzyme. And we have the DNA ligase which joins the DNA fragments by forming a phosphodiester bond between phosphate group of 5 carbon of one deoxyribose with the hydroxyl group of 3 carbon of another deoxyribose. So this is very important. So this is how this DNA ligase enzymes which will influence on the fragmented DNA. So next thing is the host cells. Here we have the host cells. The host cells are the living system, are cells in which the carrier of RNA molecules or vector can be propagated. the host cells can be a prokaryotes or eukaryotes. So this prokaryotes or eukaryotes are the microorganisms were preferred based on the application and also host nature. So once these prokaryotic cells are selected because of its fast multiplication rate and simplest nature of genome. So the easily studied genome, which is very useful for any recombinant DNA applications, rather comparing with the eukaryotes, which will be very larger and very complicated in nature. So in that case, so the easily coded DNA molecule can have more application comparing with the other eukaryotic cells. So here we have the E. coli. So this E. coli was the first organism used in DNA technology, that is the DNA recombinant DNA technology experiments. The major drawback is that it cannot perform post-translation modification. So most of us, we aware there is a post-translation transcriptional modification in eukaryotes, whereas in prokaryotes, you don't find any translational, so that is the post-translation, translational and post-transcriptional modification. So this is the major drawback if you consider the E. coli. And this is the another good diagram for plasmid insertion. So for example, so this plasmid which contains a gene for antibiotic resistance. So this is the kind of a marker, so which will enable the plasmids to resist against any other component which could be released from the host cells. And also this is the foreign DNA, this is the gene of interest, so which where in which we are going to make restrictions with same enzymes, and also this enzymes which is giving sticky in nature of DNA molecule, that is the source molecules. And once we get it, and we will do ligation process through DNA ligase enzymes, and we will be getting desired recombinant plasmids. In few experiments or many experiments, there is a possibility to obtain the mismatch or non-recombinant plasmids molecule. So only we have to select the recombinant molecules, and then we will transfer the recombinant plasmid into the bacterial cells. So this bacterial cells will try to accommodate the plasmids and also enables to replicate with its own genomic DNA, and will start to produce that particular protein from the gene of interest. So here we have another example which depicting the recombinant nature. So inserting DNA sample into a plasmid. So here we have the plasmid, that is the ECO1. So sorry, the plasmid, general plasmid, it can be a P18 or PBR 352, or we have so many types of plasmids, and those plasmids are restricted. Again, restriction steps follows here in both plasmid and the gene of interest template strand, where we are making that with using ECO1 enzymes, and then again same ligation process which happening through DNA ligase enzymes, and this will make it that particular plasmid as the recombinant. Once this is the recombinant, and we have the perfect recombinant to transfer the recombinant to the host cells. So here we have the eukaryotic host cells. So host cells, if you consider here, the human proteins should be produced in eukaryotic host cells. And also the mammalian cells possess the machinery to modify the protein to, to the active form. That is the post-translational modification. In earlier slides, we were talking about uh, the selection of host cells, that is the prokaryote or eukaryotic cells. So the prokaryotic cells which doesn't have any post transcriptional modification and translational modifications, whereas the eukaryotic cells which have the post transcriptional and post translational modifications. So here we have the example name called tissue plasminogen activator, and here we have the vectors. So these vectors are the DNA molecule which can carry foreign DNA fragment to be cloned. So those are the self-replicating into a particular host. So as usual, like generally the vectors, which should be self-replicating. That is more important, okay, because we are going to insert that particular vector molecule into the host cells, and this host cells can able to provide the sufficient environment for the vectors to replicate and produce the efficient proteins. And here we have the examples like plasmids, bacteriophages, and cosmids, and artificial chromosome vectors. So this is the important thing. So we are coming into the plasmid area, and these plasmids are the extra chromosomal, double-stranded, circular, self-replicating DNA molecule. Usually, the plasmids contribute to about 4.5% to 5% of total DNA of bacteria. So these plasmids, which we can easily found in the bacterial cells, and these plasmids can be able to make it up to 0.5 to 5% of total DNA of bacteria. So the few bacteria which can have the linear plasmids, and few can have the circular plasmids. So here we have the examples like streptomyces and other the plasmid could be PBR322 or PUC18. So this plasmids for carries a genes resistance for ampicillin and tetracycline. So that as deserve as a serve as a markers for the identification of clones carrying plasmids. So this is the another plasmid. Here we have bacteriophages. Uh, these bacteriophages are the virus. This can able to replicate within the bacteria. In case of sudden pages, the DNA gets incorporated into the bacterial chromosomes and, and remains there permanently, and which will continuously replicate it again and again, which will become a part of the bacterial cells. And again, you have some more information about, so this can take a larger DNA segments. When you are comparing with the other, that is the plasmid, for example, PUC18 or PBR322, these bacteriophages can able to accommodate larger size of DNA molecule, that is gene of interest, into their plasmids. Okay. So again, so these cells could be have more efficient expression comparing with the plasmids. For example, we have the lambda phage, that is the page lambda, and page M30. The next one we have cosmids. So these cosmids are the vectors possessing the characteristics of both plasmids and bacteriophage lambda. Those carries larger fragments of foreign DNA compared to the plasmids. So here we have artificial chromosomes vectors. So first one, like there are so many artificial chromosome vectors. So this will have more accommodation size comparing with the bacteriophage based vectors. So artificial chromosome vectors are human artificial chromosomes, yeast artificial chromosomes, bacterial artificial chromosomes. So these are the few artificial chromosomes available for our recombinant DNA technology experiments. And this can easily accommodate very larger fragments of DNA. And these are the few methods uh, which will be followed in recombinant DNA technology for gene transfer. The first one, a transformation. So this transformation procedure or the method will need to use the calcium chloride to enable the plasmids to enter into the host cells through cleaving of pores, and also it can easily obtain the plasmid DNA. And next one is the conjugation, which is the natural microbial recombination process. So in this process, we can able to push the plasmid into the host cells. So third one, we have electroporation, so which is based on the principles of high voltage electric pulses that can induce cell plasma membrane diffuse. And also there is another method, liposomes mediated gene transfer, that is the lipofiction. So this this also which needs electroporation, that is the electric voltage, to push the plasmid DNA molecule into the host cells. And we have the direct transfer of rDNA. So DNA is directly transferred into the nucleus by using microinjection or particle gun bombardment. So here we have the applications of RDNA technology. So this RDNA technology generally has, there are many applications. The first one here we have the AIDS test. So this AIDS test can be done through this RDNA technology based molecules. So if we can bring that kind of technology into this kind of diagnosis, and which will make very afford, and also the results can be easily predicted within a short time duration. And you have the diagnosis of molecular diseases like sickle cell anemia or thalassemia, familial hypercholesterolemia, and cystic fibrosis also can be easily diagnosed with this RDNA technology based molecules or the applications. And you have the prenatal diagnosis. So the prenatals are very important for any experiments which is going to diagnose the mother to check their fetus whether the fetus is nature is normal or abnormal. So this can be done through the DNA from the cells. The cells would be collected from the amniotic fluidic or chronic villi, and then we will have the RDNA technology based molecules which will confirm whether that patient has any disease, that is the genetic disorders or not. And we have the gene therapy. So this is the one of the important area in RDNA technology, because many somatic diseases are the germline based diseases, or can be cured through this gene therapy, because many diseases which should be very permanent only if you alter their genome and continuous expression of genes. So in that time only it is very possible to bring the continuous production. Okay. So for example, this can be achieved through cloning a gene into a vector that will readily to be taken up and incorporated into the genome of host cells. So for example, this is the one ADA deficiency can be successfully treated through gene therapy. There are approved gene therapy molecules. One among that is the gentisane, so which has been used by the patients, and also which is approved by the FDA. And this recombinant DNA technology also has applications in agricultural field. The first one, genetically engineered plants are can able to bring the resistance against drought and diseases. And also the second one, it can able to produce the quality food or nutrition food, are very the protein molecule, high protein molecule content in their product. So through that, the agriculture field will be became again active through this RDNA technology. And so this is the one of the another important area, industrial applications. So here we have the enzymes. In a lot of enzymes are used in the industrials to produce, maybe for example, to produce the sugars or cheese. Protein products are used as the food additives or increase nutritive value or beside importing flavor. And if you see the application in forensic medicines, and so this particular RDNA technology, which has very important role in forensic medicine, and also this can be easily used in RDNA fingerprinting, and also which can be enabled to identify the criminals, to settle the disputes of parenthood of these children. And you have the transgenesis. So this transgenesis, it's a kind of gene therapy. It can able to replace the gene in particular person, whoever is lacking that gene. Once it is transferred into the fertilized ovum, and it will be permanent and it will synthesize continuously that lacking protein or the lacking genes. And we have the gene cloning. So here the recombinant DNA molecule is transferred to the host cells. Within a host cells, it will replicate and produce dozens of identical copies. So that is the cloning. And also, once the cloned copy will have the recombinant molecule, and from that we can easily recover through purifying and analyzing, and we will check whether it is transcribed or not. And gene product is isolated and used for research and also sold commercially. So this is the complete diagrammatic representations. So here generally we have the site of cleavage, that is the plasmid vector, and site of cleavage, the gene to be cloned. So both are restricted with same restriction endonuclease enzymes, and the annealing and ligate process are initiated through DNA ligase enzymes. Once the ligation is over, the chimeric plasmid or the recombinant DNA containing plasmids we will be obtaining. And this recombinant DNA plasmids will be transferred into the bacterial cells, that is the host cells, through transformation mechanism. Now the host cell which becomes transformed bacterial cells, and through that we will have the amplifications, and the amplification of recombinant DNA molecule continuously to obtain the gene of interest. So this gene of interest again can be isolated, and so again we can be cloned into a human DNA molecules. In another step, we have the host cells. So this host cells will grow under a condition that allow expression of cloned DNA and also isolating it. And we would have confirmed that it is the exact product we needed, and we will produce it, and these products will move into the commercial market. Again, this is the same content here. We have gene for antibiotic resistance. There is a marker. This is the plasmid and foreign DNA, and the restriction is a happen, and hybridization is a happen through DNA ligations. And we have ligation process and recombinant molecule in host cells, and we will start to produce it through cloning experiments. So here we have the cloning in antibiotic medium, and from this we will purify it, and we will obtain only the recombinant molecules. So this recombinant DNA molecule also has important role in gene libraries, collection of clone DNA sequence, and each clone segment relatively small, and many separate clones are required, and libraries, genomic and chromosome specific cDNA, and so all this can be done through this rDNA technology. So these are the reference I have followed it for these presentations. And finally, so thank you so much for listening my lecture. Thank you so much.